Thank you for joining us today. I hope you can hear me loud and clear. I'm Michela Fassi, Communication Manager at Milestone, and I'll be your moderator for today's webinar. This webinar today is dedicated to the 3 key factors that impact on your tissue processing workflow. And we will discuss together with our application specialist the key elements to see when you do your tissue processing. Now, before starting, I just want to focus your attention a few seconds on this slide. Here you can see the basic function of the Zoom platform through which we are broadcasting. The meeting controls are at the bottom of your screen. You can move your mouse to make them appear. Your microphone will be muted for the entire webinar, but of course you can interact with us by typing questions in the Q&A box. So you can see the Q&A icon in the bottom of your screen. There will be a Q&A section at the end of the presentation, and we will try to answer as many questions as possible. This webinar will be recorded and will last about 40 minutes. Now, a quick presentation of today's speaker. Today's speaker is Irene Angeloni, Application Specialist in Milestone. She's graduated in Biomedical Technologies at the University of Milan. She's been working in Milestone since a few years, but she's already very prepared in tracking and tissue processing. Now, I think it's time to start, so I will switch off my presentation and I will give the floor to Irene to start the webinar. So Irene, I see that you are sharing your PowerPoint and now you have the floor.
Okay, thank you, Michela. And hello, everyone. I'm Irene Angeloni, Application Specialist here in Milestone. And today we will see together during this webinar the 3 key factors that impact on your tissue processing workflow. I would like, first of all, to start from, I think, this question that is essential to start with this webinar. Why is the laboratory workflow important? In today's dynamic healthcare landscape, anatomic pathology laboratories face several critical needs and challenges. What we see in the laboratory today is an increasing number of samples to be processed for the increased number of tests and analysis. At the same time, these analyses must be carried out in a short time while keeping costs down and also providing documentation, standardization throughout the whole process. Most importantly, quality and safety must always be the ultimate goal. According also to a study, among the things that help the change in the lab towards these needs that we have just seen is to re-engineer the workflow. So to have a better management of the lab to meet those needs, I need to re-engineer the workflow. This is essential. So the laboratory management is required to decrease costs, increase efficiency, and I want to stress this point because it's very important, with quality and safety to be of primary importance. So to sum up, the key elements that we need to look at when we talk about laboratory workflow are efficiency, quality, safety, and cost. Today, during this webinar, I would like to discuss with you these elements in 3 different steps with 3 different key factors where we connect to have an impact on the workflow. to better manage the whole workflow of the lab. These key factors are the samples processing management, the reagents management, and traceability. So let's start from the first one, that is sample processing management. That is how to better handle all the steps needed to process a sample. In particular, we are going to see how we can do it thanks to the lean workflow and also with the possibility of introducing automation in the lab. So lean workflow and process automation. What do I mean for lean workflow? Lean workflow is a process in this way. Several short runs during the day with thin samples, biopsies, small ones, and the longer process for thick and fatty tissues during the night. So basically, instead of doing in the standard way of processing all the samples together in the night, in the longer run, divide the samples and do some runs also during the day. I think that this aspect is becoming very important. I recently visited 2 different labs in 2 very different countries, one in Hamburg and one in Amman, so very different places. But at the end, they both were in the process to implement this kind of workflow. So I think that that laboratories are trying to introduce this short run during the day because they see the value of start doing dividing the samples in a different way. Of course, to do this kind of workflow, I need also to have a unit, a tissue processors, that allows me to do fast processes with also dedicated protocols with these for these different kind of samples, so for the small ones and the biopsies. And also, for example, with a dual cavity, we have the possibility to run 2 processes at the same time, so having also a higher throughput. What we can appreciate from this graphic is that this kind of organization allows for, you see, a better distribution of the workload. Why? Because we will already have during the day some samples ready to be embedded, cut, and so on, lowering the work of the morning after. And another important thing is that the unit will be used also during the day. So converting the idle time in productivity time. This is very important. Also regarding the quality, I can process very small samples with shorter protocols that are more appropriate for this kind of tissue. So regarding the quality, I have appropriate schedule for this different and fragile kind of sample. The other topic, so after the lean workflow, the other topic in the sample processing management is automation. This aspect is important, especially if we look at it related to the concept of the lean workflow that we have just seen. In fact, I'll read this. Automation supports a lean organization by improving efficiency, reducing processing errors, and enhancing safety. Also, automate a process allows to have time to invest in other important tasks. What we have talking about automation, what is our solution? We have Synergy, that is Milestone auto-embedding system. Through the auto-embedding, we skip the step of the embedding, the manual embed, and it's possible in this way to reduce time, costs, and it has very important advantages for the safety of the samples, because delicate specimens are handled only once, reducing the risk to damage or worse, to lose them. So it's very important for the safety of the samples. Regarding automation, we will see also later the automatic tracking system. Later in the webinar, we will see also this part. Now, I would like to sum up a little bit the first key factor, that is sample processing management. We have seen then that regarding efficiency, we are able, thanks to the lean workflow, to leveling the workload, to have a higher throughput. We have seen that we can also improve quality with dedicated protocols for the small samples, and that thanks to the synergy system, we can handle in a safer way the small samples, the fragile samples, and we can lower the cost. So this was the first key factor. Now, let's go into the second topic, that is reagent management. I think that it's very important to talk about reagent when talking about the laboratory workflow, also because reagents are essential for almost every analysis in the lab. Regarding the reagents, we are going to see how it's possible in this context to reduce downtime, and also the aspect more in detail of the reagent exchange. And we will see also how a better reagent management can allow for a better laboratory management, and most importantly, to have safety for the operator and samples quality. The first important way in which we can reduce the downtime is by eliminating the time where we can't use the unit. This is, for example, during the cavity cleaning. So in our processor, we can directly start with a new run as soon as the previous one is finished, avoiding these cycles of the cavity cleaning. Then, with the new tissue processor, Magnus in particular, we have the possibility to have a wax reservoir. The wax reservoir is to keep wax already melted, so that when you have to change wax, you don't have to wait the melting time. This is another kind of downtime. I don't have to wait, but I can immediately start with the run. So I can always immediately start to use the unit. Another aspect is the use of commercial tanks. And this is one of the, I think, most appreciated things when I ask to customers about feedback about our unit, is this possibility to use commercial tanks that really change the management of the reagent, because the reagent exchange, it's very easy and fast. So this is another sort of downtime, the time that you lose by changing the reagent. Here, the reagent exchange becomes very fast and very easy. I think this is important. More in details regarding the reagent exchange in terms now of frequency. We can reduce the reagents consumed by changing the reagent only when it's really needed. So this is possible through the use of what we have again in Magnus, that is an alcohol sensor. The alcohol sensor determines in a scientific way when to change the reagent. So I'm sure to change the reagent only when it's expired, or only when it's not good. We also have a complete different changing schedule for works because thanks to the use of isopropanol, we are able to change it less frequently and not every 3-4 cycles. And more regarding the quality is the possibility to have the documentation of the reagent that I'm inserting through the use of the barcode on the tank. Why this? Because in this way, I will have the information on the lot, on the reagent, also when required if facing some problems. So also this is important. Talking about, again, the use of isopropanol, we usually have isopropanol instead of cysteine. And this is an important aspect because this reagent, isopropanol, is safer. And in terms of exposure and also in terms of disposal of the reagent, the use of isopropanol is safer than other reagents such as cysteine that are very toxic. So again, in general, another particular aspect is that by avoiding the manual refill of the tank, as we see, we have the possibility to introduce commercial tank directly in the unit, so we don't have to manually refill them. We also avoid the risk of spillage. And again, this is important regarding the safety of the operator. Of course, it's essential to have also the area where you change the reagent and the area where you work that is vented to protect again the technician. So this was regarding the operator safety that it's very important. Now, regarding the samples quality, so we'll see the safety of the operator and now sample quality. This is a very important point because the reagent at the end is what, let's say, transform the tissue. So it's inevitable that the final quality, the quality that I have on the slide, it's strictly correlated to the quality of the reagent itself. I think that you can agree on this. And from our direct experience, the contamination between reagents, in particular with the water-based ones, is what gives the most problems during the evaluation of the slide. So also learning from this, we have introduced in our units a split manifold that avoids the contamination between reagents. And again, the alcohol sensor that detects eventual contamination between reagents. So very important to keep the purity of the reagent. And another important point regarding the samples quality, but we can say also the sample safety, is to have what we call safety mode. What is this? Is the fact that if something happens with the unit, with the reagent, with the contamination and so on, the samples are kept in a reagent and in a correct one. So safety for the samples that guarantees also at the end to be able to perform an analysis. So again, samples quality. Let's sum up a little bit also the second key point, because it was a lot of things. So regarding the efficiency, we can reduce downtimes with the works already melted, with the reagent exchange, and so on. With regarding quality, we always have pure reagents. We have the instruments to avoid contamination. We use reagents that are less toxic, such as isopropanol. So it's safer when you change it, when you handle it, when you dispose it. And we also, in terms of cost, reduce the reagent consumption and avoid the disposal of toxic reagent as xylene. So this was, to sum up, the second key element, that was reagent management. So third one, and also last one for today, traceability. Traceability, the third topic. Why is nowadays so important the traceability? Traceability allows us to avoid error, to increase efficiency, to easily reconstruct the history of the samples when needed, and we will see the traceability regarding the, let's say, the single cassettes, the batch of cassettes, and also in general in the monitoring of the lab. As we see also in this survey, the traceability is important also for safety. In fact, the specimen management errors is an issue that requires quality improvement efforts. And with errors, in this case, what we mean is, for example, wrong or missing code, or worse, lost cassette, this kind of error. In particular, regarding the single cassettes, also considering the rising volume of specimen that we receive, it has become vital to create automated systems that can assist in this step of tracking and managing the workflow of the specimens of the cassette. The traditional method of manually documenting samples information, test results, and so on can be time-consuming, prone to the human error, and at the end, you don't have the correct documentation available. By automating these processes through a tracking solution, instead the laboratory can increase the efficiency and reduce the risk of errors, and having also a complete and accurate documentation. So I think that pathology labs need a built-in solution that is faster, that is accurate, that automatically provides sample tracking from the start to the finish of the step. In this video, I would like to show you the functioning of T-Tracker, that is our automatic basket scanner, so our automatic tracking solution. And I want to show you this video because it shows what we call the dual sensor technology. That is the counting of the cassettes thanks to the laser sensor, and in a second step, the reading of the code thanks to the camera sensor. Why I'm telling you this? Because thanks to this, we are able to detect if errors are present, if we have missing codes. And in this way, we have a reliable scan, we have the documentation, and also regarding, in general, the safety of the sample, we detect if something is wrong. We have seen the traceability in the step of the cassette with the tracking solution. But to have a complete documentation, it's important to track the specimen from during its entire journey. So from the operating room to the crossing to the processing step. So the entire path of the sample. traceability of the sample during processing. Then very important regarding especially what we are seeing in the context of the laboratory workflow is the constant control of the units present in the lab, the communication with the LIS system, and the possibility to take immediately action in case of unattended problems. Our solution to meet all these needs is Milewatch. Milewatch is our software that allows to track the path of the samples, so of the single, let's say, case ID, the single cassette, along its entire journey, and at the same time, it's also monitoring the system because we can monitor all the Milestone's units in the lab, have notification, see the status, see what's going on, see the alarms, and so on. So Milewatch tracking and monitoring system. Monitoring, it is connected to the unit. Here I have a slide that represents a little bit all the connection that Milewatch has with our Milestone units, and it's able to communicate and exchange data with the unit, starting from the transport of the sample to the pathology lab, to the grossing step, up to the tissue processor. This was in general Milewatch, but in this particular context of the laboratory workflow, I would like to talk to you about this tool that I find particularly useful to support also the change in the workflow of the lab, that is the real-time processing. Because thanks to this tool, you can see how many samples you have in a specific phase. So the lab manager can see how many samples you have to cut, you have to embed, you have already processed, you have to process, and so on. In this way, it's, I think, a nice possibility to have a better visualization of the samples that you have, to have a better distribution of the workload, and also a management of the unit. How many embedding station, how many technicians for the cut, and so on. So I think it's nice. To sum up, also the third point, we are almost at the end, traceability. If the laboratory wants to gain efficiency and implement a better workflow, it's important to have the correct tracking and monitoring system that can sustain this change, that can document, so documentation, all these steps, and also considering the safety of the samples in terms of traceability. We always know in which step the sample is. We can detect if errors with the codes are present, and with the automatic tracking system, we avoid also the time-consuming procedure of the manual scanning, and we can use this time for something useful. We have arrived now at the end of the webinar. To conclude, let's see once more the 3 key factors where we have room for action to improve the laboratory workflow. The management of the samples with the idea of the implementation of the lean workflow with the processing of some samples during the day, and also by introducing automation with the auto-embedding system, automation that we find also with the tracking line to automatically track the cassette, the reagent management with a safer and especially easier way to handle the reagents, and the traceability. Traceability, that is what can, I think, sustain the whole change in the workflow and to really improve the efficiency. So thank you very much for having listened to me. Now I'm ready for all your questions.
Thank you, Irene, for your nice explanation. I have already collected some questions, so I give you time to take off your PowerPoint. And in the meantime, of course, all the attendees are invited to type your question in the Q&A box so I can ask the question directly to Irene. So Irene, the first question for you is this one. We have an attendee who says, I did not understand why you eliminate the cavity cleaning. Is it only in Magnus?
Okay, thank you, Michela. Yes, I didn't go in detail of the cavity cleaning, but what is the functioning? In our processor, so in Magnus, but also in the Logos, as you can see, we have two different cavities. One cavity is dedicated to all the reagents such as formalin, ethanol, and so on, while one cavity is dedicated only to wax. So in particular, in the case of Magnus, the rack, once it has finished all the steps in the main cavity, thanks to the robotic arms, it automatically moves in the wax cavity. So wax it is not moved, and for this reason, since we don't move the wax, we also don't have at the end of the run to clean the cavity because wax remains in that cavity. With Logos Evo, sorry, going this part, it's the same concept. We have a cavity for all the reagents and a cavity for dedicated only to wax, and we have the possibility, when the technician is present, to manually move the rack from this cavity when all the steps of ethanol, isopropanol, and so on are finished, move this rack into the wax cavity. So again, also in this case, wax is not moved, and you don't have to perform the cavity cleaning at the end. So you can immediately start with a run.
Okay, thank you, Irene. Let's move to a second question. Why is it better to use the alcohol sensor? You have to change the reagent anyway. So maybe it's a bit tricky, but let's try.
Yes, you have to change the reagent anyway, but we have seen from our experience that through the use of the alcohol sensor, the customer have the possibility to use more the reagent. So what we usually do is to change the reagents based on a number of cycles and number of cassettes that derives from our test, but is in any case a number that is not true for everyone, because it depends on the specific reagent, on the kind of samples, on the number of samples, and so on. With the alcohol sensor in Magnus, instead, this calculation on the expiration is something scientific. So you have the percentage of purity of the reagent, and in this way I can change it less frequently and only when it is really expired. So for this reason, it's better to use it, and you can use a little bit more the reagents.
Okay. Now a question about traceability. Does the scanner, so I think he refers to the T-tracker, does the T-tracker only work with the square racks, or is it possible also with the Magnus rack?
Okay, no, T-tracker works with every kind of basket, so with the squared one, but also with the Magnus. I have here these accessories. That is the tool that you need to insert the rack in the unit, and this is, for example, the one for Magnus and for Logos Evo, but you have also the one for the squared rack. So it can manage the squared one, but also the circular one that are our racks, and also the semi-circular rack from Excelsior. So yes, also our rack.
Thank you again. Now I have a question which refers to the split manifold. What do you mean by water-based?
Okay, with the water-based reagent, we mean let's say for us, the first two reagents that are formalin and alcohol 70, because formalin is made starting from water, and also the alcohol 70 has an high, let's say, high percentage of water. So the first two reagents, formalin and alcohol 70, are water-based reagent. And with the split manifold, we mean that inside, in the drawer, we have, now I show you a little bit also with the camera. In this way, okay. The first two positions for the first two tanks here have a different circuit from all the others, so that we avoid the contamination between the first two reagents that are water-based, formalin and alcohol 70, and all the other that are instead alcohol-based reagent. Alcohol-based reagent meaning ethanol, isopropanol, and so on. It's important because if you have the water contamination, the slides have like a sort of white area that really it's difficult then to do the analysis of the slide. So I think that trying to avoid the water contamination is the contamination between reagents in general is something important.
Okay, thank you again. And linked to your answer, we have another question again regarding the split manifold. Is that available in Logos as well?
So the split manifold is present in Logos Evo. That is this one that I have here together with Magnus. So in Magnus and the Logos Evo, we have the split manifold that are, I think Logos Evo is the last one, and Magnus some years ago. From Magnus on, we have this kind of configuration of the manifold.
Thank you again. I think we have time for one final question, and all other questions have been answered. So one final question for you, Irene, for the moment.
Yes.
I do understand the concept of lean workflow, but I think that you need to have a lot of small samples to be able to implement it. What can we say about it?
Okay, thank you for this question. Yes, so let's say that you don't require a lot of sample, and this is also, I think, an important concept on the lean workflow. You can adopt this kind of workflow also if you have a few samples. What do I mean? That, for example, I have only 20 small samples biopsies during my daily routine. I can run only this small batch of samples with one run during the day and then all the other during the night. Of course, if I have a lot of small samples with Magnus, with our tissue processors, you have the possibility also to manage this high number of samples by doing several runs during the day. But also, if you have few samples, if you have just the 20 small samples, you can start by implementing this lean workflow by processing them in a separate run during the day. So no matter if they are 20 or 210, you can anyway, let's say, separate them and start to process them during the day with a linear workflow. Thank you.
Thank you once again, Irene, for your time and your explanation. We have satisfied all the questions for today. So let me share one final slide with you. Here you can see our contacts. So in case you need any additional information, in case you want to plan and arrange a virtual demo customized on your need, on the need of your lab, please don't hesitate to write us, to contact us. Tomorrow you will receive the webinar recording, so you can rewatch your webinar when you need. One final information for you. We already have another educational webinar already planned for next month. It will regard the grossing stations, so registration will open very soon. And of course, I invite you to follow us on our social networks, LinkedIn, Facebook, and YouTube, to stay always up to date with our news and events. So thank you once again for watching and participating, and we hope to see you all next time. Thank you and goodbye.